Hello everyone, I'm Alejandro Montenegro and I'm the Executive Editor for Cold Spring Harbor Protocols. I'm joined today by Alejandro Sanchez Alvarado, who is the President and Chief Scientific Officer at the Stowers Institute for Medical Research, to talk a little bit about the research and the work that we'll present at the 87th Cold Spring Harbor Symposium, which this year focuses on stem cells. Thank you very much for taking the time.
No, thank you very much for having me, Alejandro.
So, work in your lab seeks to uncover the molecular mechanisms responsible for regeneration and development.
Mm-hmm.
And for this, you've established the use of a very interesting model system, the Planarian flatworm.
Mm-hmm.
So I was wondering if you could tell us a little bit about this system and some of its advantages.
Yeah, happy to. I mean, so Planarians were selected some 20 years ago or so, maybe a little longer than that, for study in my lab. Because of all the animals that I knew of the time, this one in particular exaggerated in a great way the process of regeneration. These animals can be cut and sliced and diced in a number of ways, and every time that you do this, the animals are capable of launching a regenerative response and restoring all the missing body parts. You can take a small fragment out of the animal and watch that little fragment reorganize itself and produce a completely, well-proportioned animal. And so this type of whole animal regeneration, partial regeneration, reconstruction and so forth were so evidently manifested by this animal that I figured that being the wild type phenotype, if we can perturb it genetically, imagine all the phenotypes that would emerge. And that's one of the main reasons for selecting the animal.
Mm-hmm. So you've also identified somatic stem cells in planaria. Are these the key cells for the regeneration capacity of this system?
Yeah, so they were known. The existence of these cells were known. Another reason for selecting the animal, they were originally described in the late 1800s by a number of scientists, among them Harry Randolph and Bardeen and Becher and others, and including T.H. Morgan was aware of the existence of these undifferentiated cells. And for a long time, it's been believed that they are the main cellular agents of regeneration. And the reason why that's part of the understanding of planarians to this day comes from those experiments done by Bardeen and Becher, where they essentially irradiated the animals to get rid of the neoblasts, which are the only stem cells, the only cells that actually divide in the animal. Mm-hmm. And what they noticed is that when those animals that were irradiated were devoid of the stem cells, the animals would actually die and were incapable of regenerating. So for many years, people had imagined that these cells, these undifferentiated somatic, very likely pluripotent stem cells, were the key cellular agents of regeneration in planarians.
In your presentation, you'll discuss insights derived from single-cell and spatial transcriptomics to help understand pluripotency and regeneration in planaria. Yeah. I was wondering if you could tell us a little bit about what those insights are.
Yeah, I mean, a number of insights have emerged from these studies. The first one that emerged very early on in the process was by applying these new methods from microarrays to next generation sequencing to single cell sequencing, particularly to the stem cells, that while they all appear to be morphologically quite similar to each other, molecularly they're fairly heterogeneous. And those cells that are most undifferentiated are also themselves transcriptionally quite heterogeneous, to the point where we were able to identify at least 12 different types of neoblasts based on their transcriptional profiles, all undifferentiated. That was a big surprise, suggesting that these cells may be occupying a diversity of transcriptional states. And so I was expecting it to be much simpler, but nature always pushes back whenever you push it back. That's one insight that emerged from that. But the other insight that is emerging as well in our understanding of regeneration in planarians and perhaps in animals in general is the fact that also the differentiated cells are not terminally differentiated, to the point that when homeostasis is perturbed by injury or by amputation to launch a regenerative response, many of these so-called terminally differentiated cells will actually change their transcriptional profile as a response to injury to create an ecosystem that will allow the neoblasts and other cells to actually launch this regenerative response. So these two notions that the stem cells were unique and they have a particular profile, and that the differentiated tissues were essentially already differentiated to the end, that is no longer the way I think about regeneration in planarians, and I'm beginning to extrapolate those ideas to understanding regeneration in other systems as well.
In your work, you highlight the role of certain microenvironments to power the whole body regeneration. Can you tell us a little about
Yeah, so it turns out, at least with the most recent data that we have accumulated, that there is not necessarily a uniquely defined niche where the stem cells reside. It appears as if there's a large number of such niches, and that those niches, all of them appear to be perturbed whenever amputation or loss of homeostasis it occurs. So it appears as if the niche is essentially composed of the friends that the neoblasts make along the way to regeneration. So we find close association of these neoblasts with things like muscle, things like parenchymal cells, things like gut cells. And if you perturb any of those components, you actually see consequences in the ability to regenerate. So now I'm beginning to think that this deterministic thinking about the cell, the stem cell occupying Eden or nirvana surrounded by well-defined differentiated cells, while appropriate for some systems like the gonads of Drosophila or even C. elegans, it may not be appropriate for understanding how a pluripotent adult somatic stem cell actually retains its homeostasis, its ability to respond to changes and not grow out of control. So I think that's an interesting and important insight that's emerging from these studies.
Mm-hmm. So what do you think, or what's known about why only certain metazoans can regenerate fully after amputation? What has been lost in the ones that can't, or what is a characteristic of those that can?
Yeah, I think that's a problem that is probably going to succumb to a scientific investigation in the next couple of decades, if not sooner. And the reason why I say that is because there's now a relatively large community of people working on a diverse number of animals trying to exactly answer this question. So one of the most puzzling characteristics of animal regeneration is that it is broadly but unevenly distributed. You find it in many animals, in many phyla, but you can find two animals that are closely related. One has it, the other one doesn't have it. It's not entirely clear if this is something that occurred once in evolution and it got distributed in some interesting way across the metazoan tree of life, or whether the ability to regenerate emerged independently in different animals, or it's a combination of the two. But the molecular tools and the cellular tools that are available to us today are going to allow us to address this question and possibly come up with an answer as to why is it that you and I cannot regenerate a limb, or why is it that salamanders can regenerate a limb, or why is it, for example, that the axolotl cannot regenerate a lens, but the newt can. And these are really closely related organisms, right? So it's still a mystery. And I think it's one that is finally, after hundreds of years of study in regeneration, might actually succumb. And it's not clear to me to what extent things are lost because of drift, or things were lost because of negative selection, or things were kept because of positive selection. All that still remains to be addressed. But I get the sense that the work from my lab and the labs of many other scientists around the world are beginning to chisel away at this puzzle. And it's going to come into focus more and more in the decades ahead. So I'm very excited about it. I just wish I were younger so I could be a graduate student again and be part of that. But I think it'll be quite interesting to see how it unfolds.
So how can work in quote-unquote simple organisms can help us better understand development and regeneration in more complex systems?
That's a really good and fair question. We are trying to understand our own biology as human beings. We are afflicted by a number of maladies that we don't have answers to yet. And so for the past 100 years or so, since the advent of genetics, it became apparent that in order for us to understand what afflicts us, we need to understand mechanisms. And sometimes those mechanisms are really hidden in the human, existence, right? So what do? Well, you go to systems that exaggerate certain attributes where you can actually molecularly and genetically dissect them. So just as we learn about a host of different diseases from yeast and Drosophila and Tetrahymena, all of these really relatively simple organisms to inform our biology, it's not too much of a leap of faith to imagine that by looking at animals that regenerate, we're going to be able to extract first principles that will be applicable for us to understand what do we need in order to activate or introduce or reawaken some of these processes that may allow us to, restore axonal growth, for example, for spinal cord that are severed, or perhaps a restoration of neurons in the brain as a consequence of Alzheimer's or ALS or any of these other maladies. So I do think that the studies of these systems is going to allow us to uncover those first principles, and like we've done in the past in developmental biology and other fields, those principles will be applicable to our own biology. Because, as you know, I didn't have to work out this way, but we do share common ancestry with all of these different organisms. That's the way it worked out. We've sequenced enough genomes to know that we really are closely related to almost every other metazoan that exists on this planet. So who is to say that the secrets and solutions to many of the problems that afflict us today are not already solved by animals out there? And they're just waiting for us to knock on their doors and say, hey, could you share with us how you solved that problem? So I think that's one of the key reasons why all of these systems will eventually be extremely informative to our own condition.
Great. So thank you very much for this discussion.
Thank you.
It has been great having you at the lab.
Well, thank you very much, Alejandro. It's been a pleasure chatting with you as well.
Yeah. And thank you all for watching.